Hi everyone, in this video we will look at how action potentials are conducted along an axon, including the importance of the refractory period and the factors that affect the speed of conduction. The refractory period is the short time that follows each action potential when the membrane cannot be depolarized, and so another action potential cannot yet be initiated. This is due to the nature of sodium ion channels. Once closed, they cannot be immediately opened again and are in a deactivated state for a short time. During this time, the resting potential is also re-established by the sodium-potassium pump. Once resting potential is restored, another action potential could be generated. The refractory period is essential in ensuring that the action potentials are only conducted in one direction, as we'll see on the next slide. It also allows for them to be discrete and non-overlapping, and it also limits their frequency. As frequency of action potentials increases with the intensity of the stimulus, this means there's a maximum stimulus intensity to which we are able to respond. In our earlier videos, we've seen how positively charged sodium ions diffuse into the axon during depolarization. These charged ions repel one another inside the axon, causing adjacent parts of the membrane to also become depolarized, and so the action potential is propagated. The membrane behind the action potential is still hyperpolarized in the refractory period, and so cannot be depolarized. This is how action potentials are conducted in one direction only. In the absence of a myelin sheath, we call this an unmyelinated axon. This process of depolarization, leading to the depolarization of adjacent membrane, continues down the entire length of the axon. However, in some neurons, their axons are surrounded by a myelin sheath, and in these myelinated axons, the action potentials can be transmitted much faster as depolarization only occurs at the nodes of Ranvier. This is called saltatory conduction. The myelin sheath is a fatty layer that provides electrical insulation to the parts of the membrane it covers. This means that no depolarization occurs at these parts of the membrane. Where depolarization does occur, the sodium ions that diffuse in repel one another, leading to depolarization at the next node of Ranvier. As depolarization only occurs at the nodes of Ranvier, the action potential is conducted much faster. It also means that less energy is required, as ATP is only needed for the sodium-potassium pump at the nodes, rather than down the entire length of the membrane. The speed of conductance can be found by dividing the length of the pathway by the time taken for the action potential to be propagated down its length. Three factors can impact this. As we've already seen, in saltatory conduction, action potentials are conducted faster along myelinated axons. Secondly, the diameter of the axon also affects the speed of conductance, with wider axons providing less resistance and therefore faster transmission. The final variable is temperature. An increase in temperature enables faster diffusion of the ions. However, this only applies up to around 40 degrees C, as beyond this the protein channels will start to denature. This is the last of our videos on the action potential. In the next video, we'll find out how neurons interact at a synapse.